Welcome back to the HIV RNA Test Guide podcast. Your trusted source for reliable information on HIV testing, prevention, and treatment.
Absolutely.
And ready to explore something amazing with us.
I am.
We're going to take a deep dive into all the latest research into developing a vaccine for HIV.
It's such a fascinating field.
It is. Our goal here is to give you a clear understanding of where the research is headed, even if you don't have a scientific background.
Right.
We're talking cutting-edge stuff here, the kind that could actually maybe end the HIV/AIDS epidemic.
It really could.
Yeah. So the article that we're looking at for this deep dive is called Emerging Topics in HIV Vaccine Development.
Great article.
Yeah, and it really is. It's a great overview of what's going on in the field. So I guess to start off, one of the things that the article talks about is broadly neutralizing antibodies.
bNAbs.
Yeah, those sound pretty intense.
They are, yeah. They're really the elite forces of your immune system.
So how are they different from regular antibodies?
That's a great question. So HIV is really tricky. It's a master of disguise. It's constantly mutating. So it's like trying to grab smoke. Regular antibodies just can't catch it.
Yeah.
But broadly neutralizing antibodies target those really tiny parts of the virus that can't change. The bits that HIV needs to survive.
Okay.
Yeah.
So how do scientists even find those tiny, unchanging bits?
It's really incredible. They use something called structural biology. One technique is cryo-electron microscopy.
Cryo-EM?
Yeah, cryo-EM.
Wow.
And that allows them to see the virus in incredible detail.
Wow.
Down to the arrangement of atoms.
Oh, wow. So they're basically creating a 3D map of the virus.
Exactly.
Wow.
With that map, they can pinpoint those weak spots and design vaccines that teach the immune system, hey, this is what you need to target.
That's amazing.
Yeah.
The article mentioned that there was a trial where 97% of participants developed these targeted responses.
Yeah.
And that trial was called IEVI-G001, and it used a strategy called germline targeting.
Right.
What is that?
So essentially, they're training the immune system from the very beginning to make those bNAbs, like a boot camp for your immune cells.
Okay. I'm picturing tiny soldiers doing push-ups.
Exactly.
So are bNAbs the only weapon in the arsenal?
No, not at all. Scientists are developing completely new ways to deliver these vaccines, kind of like upgrading from snail mail to high-speed internet.
Speaking in high speed, we saw how quickly mRNA vaccines were developed for COVID-19.
Right.
Is that technology being used for HIV too?
Yeah, definitely. So Moderna, for example, is running trials with mRNA vaccines that specifically target HIV.
Yeah.
And the advantage here is speed. So the immune system learns to recognize HIV much faster. Plus, they can easily tweak the vaccine if the virus mutates.
That's really interesting. The article also mentioned something called viral vectors. What are those?
So think of viral vectors like Trojan horses.
Okay.
Scientists take harmless viruses, they get rid of their ability to cause disease, and then they use them to deliver HIV antigens right into the body.
Oh, that's so clever. And these viral vectors are constantly being improved.
Yeah, they're fine-tuning them to trigger even better immune responses. For example, the HVTN 702 trial used modified versions of viruses called ADO26 and MVA as vectors.
Okay, so it's like they're upgrading those delivery trucks to make them more stealthy and efficient.
Exactly.
Yeah. So while they're working on that delivery, they're also kind of becoming like master architects, designing those actual vaccine components.
Right.
And this next section I think is super interesting. It's called high-tech design.
Yeah.
It sounds like something straight out of a sci-fi movie.
It kind of does.
Yeah.
It's pretty futuristic.
Yeah. So, we talked before about HIV's, constantly changing outer shell.
Right.
What are scientists doing to kind of overcome that challenge?
That is one of the biggest challenges in HIV vaccine design, right?
Yeah.
You're trying to design something that can lock onto this shell even as it's changing.
Right.
So one of the techniques they're using is something called envelope trimer stabilization. What?
Yeah. So imagine the outer shell of HIV is covered in spikes.
Okay.
Those spikes are really wobbly and unstable, which makes it hard for the immune system to get a good grip.
Right.
Scientists are engineering these spikes so that they stay in a fixed, recognizable form.
Oh, okay. So it's like creating a stable target for the immune system to practice on.
Exactly.
Okay.
And remember that cryo-EM we talked about earlier?
Yeah.
It plays a key role here too.
Mm-hmm.
So by seeing those spikes in atomic detail, they can design them to be even more stable and recognizable.
Wow. That's really impressive.
Yeah, it is.
But, all this talk about designing spikes and mapping viruses, it makes it sound like scientists are doing this all by hand.
Right.
What about artificial intelligence? Is that playing a role?
Definitely. AI is a huge part of HIV vaccine development, especially in something called computational design.
Okay.
Imagine you could create and test thousands of vaccine designs in a computer before even stepping into a lab. That's what AI allows scientists to do. Wow, so it's like having this virtual lab where they can run simulations.
Yeah.
And predict which vaccine designs will work best.
Exactly.
That's awesome.
Yeah.
And one example is the Mosaic trial, which used a computationally designed vaccine that targeted multiple HIV strains at once.
Right.
Okay, so we've talked about, all this amazing technology, these great designs.
Right.
But what about making sure that the immune system is actually ready to fight?
Right. It's not just about the vaccine itself. It's about making sure that the body's natural defenses are ready to respond.
Yeah. How do they do that?
So that's where the concept of innate immunity and adjuvants comes in. Think of it like this. Your immune system has an alarm system.
Okay.
And when it detects a threat, it needs to sound that alarm to call in the reinforcements.
Right.
So adjuvants are like the alarm bells.
Okay.
They're special ingredients that get added to vaccines to make them more potent, more effective. And they basically shout, hey, immune system, we got a problem here.
So what kind of adjuvants are they using for HIV vaccines?
So one promising type is called TLR agonists. And these molecules, they activate very specific receptors on immune cells. And it's kind of like flipping a switch that tells those cells to go into overdrive.
Okay, so it's like giving the immune system a megaphone and a shot of espresso.
Exactly.
Okay, so they're really pulling out all the stops here.
They are.
But, we've talked a lot about antibodies.
Right.
But aren't those just one part of the immune system's defense force?
They are.
Yeah. What about T cells?
Right.
Don't those play a role too?
They do. T cells are like the special forces. They're trained to seek and destroy infected cells.
Right.
So scientists are working on vaccines that specifically activate these T cells to target and eliminate HIV-infected cells.
So it's like we're building this whole army with different units specializing in different tasks.
Right.
Are there any parts of the body besides the bloodstream that they're focusing on?
So remember how HIV enters the body through mucosal tissues, like in the gut or genital tract?
Right.
Scientists are exploring this really interesting concept called mucosal vaccination.
Oh, okay. So what is that?
Imagine you're setting up defenses right at the front lines, where HIV first tries to enter.
Ugh.
Instead of waiting for the virus to get into the bloodstream, you're trying to stop it right at the source.
Okay.
Yeah.
That makes sense. So it's like building a wall at the border instead of waiting for the invaders to get into the city.
Exactly.
And what kind of techniques are they using for this?
They're using a variety of different approaches, from specialized delivery systems to nanoparticles that can carry antigens right to those mucosal tissues.
So it's like building tiny missiles to target those entry points.
Right.
It's like science fiction.
It kind of is, yeah.
Okay.
Yeah.
So we've talked about, prevention, but what about finding a cure for HIV?
Right. That's ultimately the goal. And researchers have been working towards this for decades.
Yeah. So where do things stand with that right now? Are there any promising leads?
Definitely. One avenue that's gaining a lot of attention is the development of therapeutic vaccines.
Oh, therapeutic vaccines. How are those different from the preventive vaccines we've been talking about?
So preventive vaccines aim to stop you from getting infected in the first place, but therapeutic vaccines are designed to control or even eliminate the virus that's already in the body.
So it's like a cleanup crew for HIV.
Exactly.
Okay.
So they're designed to target those reservoirs, those hiding places in the body where HIV can lie dormant and evade the immune system.
And how do those therapeutic vaccines actually work?
So they often work in tandem with something called latency reversing agents. Imagine these agents as like smoke bombs that flush the virus out of its hiding places, making it visible to the immune system.
Oh.
Yeah.
That's really clever.
Yeah.
So they're smoking out the virus and then hitting it with this targeted immune response.
Yeah.
What about gene editing? Isn't that the technology that can actually, rewrite our DNA? Could that be used to cure HIV?
Gene editing is definitely a game changer, and it has huge potential for treating and even potentially curing a wide range of diseases, including HIV.
Okay, so can you remind me what gene editing is?
So imagine you have a tiny pair of scissors that can precisely cut and paste DNA. That's essentially what gene editing tools like CRISPR-Cas9 allow scientists to do.
Oh, wow.
Yeah.
And how are they using that for HIV?
So one approach involves modifying a gene called CCR5, which is like a doorway that HIV uses to enter cells.
Okay.
By disabling this gene, scientists can make cells resistant to HIV infection. Another approach involves inserting anti-HIV genes directly into cells, turning them into mini factories that churn out anti-HIV proteins.
Wow. So they're like rewiring those cells to fight HIV.
It is pretty incredible.
Yeah.
The research is still in its early stages, but the results so far are really promising.
That's amazing. So we've talked about prevention, we've talked about treatment, and even some potential cures.
Right.
But wouldn't it be even better if we could combine different approaches for even more protection?
Absolutely. Combining strategies is definitely at the forefront of HIV prevention research. For example, we already have KPS,
Right.
which is a medication that can dramatically reduce the risk of HIV infection.
Right. Pre-P has been a game changer.
Totally. And now scientists are exploring ways to combine pre-Pbis with vaccines for even greater protection. It's like having a double layer of defense.
That makes perfect sense. Are there any trials looking at this combination approach?
Yeah, there are. The pre-PVAC trial, for example, is evaluating how well these two strategies work together to prevent HIV infection.
Wow.
And the hope is that this combined approach will be even more effective than either strategy alone.
That's really exciting.
Yeah.
It sounds like HIV prevention is becoming more and more effective.
It really is.
But even with all these great advancements, we need to make sure everyone has access to them, right?
Absolutely. Equity and access are critical. We saw during the COVID-19 pandemic how disparities in access to prevention and treatment can have such devastating consequences. We can't let the same thing happen with HIV.
So what's being done to ensure equitable access to these new HIV prevention tools?
There are some amazing initiatives working to address this. For example, the HIV Vaccine Trials Network, HVTN, is committed to ensuring equitable distribution, diverse trial participation, and strong community engagement in HIV vaccine research.
That's great to hear.
Yeah.
So it sounds like they're really committed to making sure these advancements benefit everyone.
Absolutely. It's a global challenge that requires a global solution. And speaking of challenging, there's one more topic I think we should touch on: human challenge trials.
Okay. I've heard about those. They've been pretty controversial, right?
They have. So human challenge trials involve intentionally exposing healthy volunteers to the virus.
Oh, wow.
Which obviously raises some ethical concerns.
Right. I can see why that would be controversial.
Yeah.
So why would scientists even consider doing that?
The main advantage is speed.
Okay.
By intentionally infecting volunteers, scientists can get much faster results, potentially shaving years off the vaccine development process.
Right.
But there are significant risks involved, even with the best medical care.
Right. So it's this balancing act between those potential benefits and the potential risks.
Exactly. And that's why there's so much debate. It's a really complex ethical issue.
Yeah.
There are no easy answers.
For sure. Okay. So last but not least, let's talk about the gut microbiome.
Oh, yes.
It seems like this mysterious world of bacteria that lives in our intestines is connected to everything these days.
It really is.
Yeah.
It's so complex, and we're just beginning to understand its influence on our health, including how it affects our immune systems.
Right. So what's the connection between the gut microbiome and HIV vaccines?
So research suggests that the composition of our gut microbiome could actually influence how well our bodies respond to HIV vaccines.
Okay, so having a healthy gut microbiome could potentially make those vaccines more effective.
That's the idea. Scientists are exploring this connection to see if we can manipulate the gut microbiome to enhance vaccine efficacy.
Okay. So could we use things like prebiotics or probiotics to actually boost that immune response?
That's one possibility. But we need more research to really understand how it all works and whether it's a viable strategy.
Okay. Well, this has already been a really fascinating deep dive.
Yeah.
It's incredible to see how far HIV vaccine research has come.
I agree. It's a really exciting time for the field.
But before we get too carried away, it's important to acknowledge that there have been some setbacks along the way.
Right. Not every trial has been successful. For example, the mosaic trial, which was testing a very promising mosaic vaccine, it didn't meet its primary endpoints.
Yeah, I remember that. It was a big disappointment.
Yeah.
But it sounds like even these setbacks can be valuable.
They can. Even when a trial doesn't achieve its intended goal, scientists learn a lot from the data.
Right.
And it helps them to refine their approach and develop even better vaccines in the future.
So it's all part of the process.
It is. And despite the challenges, the field is moving forward at such a rapid pace.
Yeah.
There's a real sense of optimism that we haven't seen before.
That's great to hear. It really sounds like we're on the cusp of some truly groundbreaking discoveries.
I think we are.
Well, this has been an amazing overview of the current state of HIV vaccine development, but we've only just scratched the surface.
There's a lot more to explore.
Yeah. So join us for part 2 of this deep dive as we delve even deeper into some of these amazing approaches and really kind of explore what the future holds for HIV vaccine development.
Welcome back.
We covered so much ground in the last part.
Did.
From, those powerful bNAbs to the role of the gut microbiome.
Right.
I'd love to kind of pick up where we left off and go a little bit deeper into the concept of therapeutic vaccines.
Yeah, let's do it.
So we talked about how they're designed to, control or eliminate the virus that's already in the body. But how do they actually do that?
Well, remember those viral reservoirs we discussed? Those sneaky hiding places where HIV can kind of evade the immune system?
Right.
Therapeutic vaccines are all about targeting those reservoirs.
So it's like a seek and destroy mission for HIV.
Exactly. But before they can destroy those hidden viruses, they gotta flush them out into the open.
Okay.
And that's where latency reversing agents come in.
Okay.
They act like those noisemakers you might use to scare birds away.
Right.
Except in this case, they're scaring the virus out of hiding.
That's a great analogy.
So the latency reversing agents flush out the virus, and then the therapeutic vaccine comes in and trains the immune system to attack it.
It's like a 1-2 punch.
Exactly.
The article mentioned that there are, different types of therapeutic vaccines.
Right.
Can you give us some examples?
Sure. One type that's showing a lot of promise is called a dendritic cell vaccine.
Dendritic cells.
Yep.
Those sound familiar.
So they're specialized immune cells that act like teachers showing other immune cells what to target.
Okay.
Yeah.
So with a dendritic cell vaccine, they're basically giving the immune system, like a personalized lesson on how to fight HIV.
Exactly. Scientists take these dendritic cells from a patient's blood, expose them to HIV in the lab, and then they inject those trained cells back into the patient.
Wow.
It's like giving the immune system a crash course in HIV recognition.
That's amazing. So they're harnessing the power of the patient's own immune system to fight the virus.
Precisely. And another exciting approach is using mRNA technology to create therapeutic vaccines.
Like the mRNA vaccines that were so effective against COVID.
Exactly. In this case, they're designing mRNA sequences that will instruct cells to produce HIV proteins. So they're essentially turning those cells into tiny vaccine factories inside the body.
That's pretty mind-blowing. It seems like we're really pushing the boundaries with, vaccine technology.
We are, and while there's still a long way to go, the progress is really encouraging.
Yeah, it's exciting to think that we might one day be able to not only prevent HIV, but actually treat and cure people who are already living with it.
It's a goal worth striving for.
Definitely. And speaking of goals, let's switch gears for a moment and talk about a topic that's a little bit more controversial, those human challenge trials.
Right.
Yeah, this sparked a lot of debate. Yeah.
Now, they have, for those who might not be familiar, in a human challenge trial, researchers intentionally expose healthy volunteers to the virus.
Wow, so instead of waiting for people to naturally become infected, they're deliberately infecting them.
That's right. It creates a controlled environment where they can closely monitor the infection process and test a potential vaccine or treatment.
I can see how that could speed up the research, but it seems incredibly risky.
It is, and that's why it's such a complex ethical issue.
What are some of the ethical concerns?
Well, the most obvious one is the potential harm to the volunteers.
Right.
Even though RT is very effective, there are still risks with HIV infection, including potential long-term health consequences.
Right.
And then there's the issue of informed consent. Can someone truly consent when they're being asked to expose themselves to a life-threatening virus?
That's a tough one.
It's a topic that needs a lot of careful consideration, weighing the benefits against the risks, and ultimately it's a decision that has to be made on a case-by-case basis with, a strong emphasis on ethical oversight and the well-being of the volunteers.
For sure. Okay, let's lighten the mood a bit and talk about something a little less controversial, the gut microbiome.
The gut microbiome.
Yeah, it's amazing how much we're learning about this whole ecosystem of bacteria living in our gut.
It's like a hidden world inside us.
Right, and it seems like it's linked to just about everything these days.
It really does.
From digestion to our immune system.
Absolutely. And when it comes to HIV vaccines, there's growing evidence that the gut microbiome could play a role in how our bodies respond.
So having a healthy gut microbiome could potentially make HIV vaccines more effective.
That's the idea. There's a lot of excitement about this connection, and scientists are looking at ways to manipulate the gut microbiome to boost vaccine responses.
So we could potentially use things like probiotics or prebiotics to enhance the effects of the vaccines.
That's one possibility, but still early. We need more studies to understand the complex interplay between the gut microbiome, the immune system, and HIV vaccines.
Yeah, it's fascinating to think that something as, seemingly simple as the bacteria in our gut could have such a big impact on our health and our ability to fight off diseases like HIV.
It is. It just goes to show how interconnected our bodies are and how much we still have to learn about our immune system.
This deep dive has been a real eye-opener. We've explored so many different approaches to HIV vaccine development.
It's been an incredible journey so far.
From high-tech design to the power of our gut bacteria.
And we're not done yet.
That's right. There's still so much more to explore. Welcome back to the final part of our deep dive into HIV vaccine development. It's been quite a journey so far.
We've covered a lot, but saved some of the most mind-blowing stuff for last.
I'm especially fascinated by gene editing. It feels very sci-fi.
It does, doesn't it? But it's becoming a reality, and it really has the potential to change how we approach, HIV treatment and prevention.
We talked about gene editing a bit in the last part. Can you remind me how it's being used for HIV?
Sure, remember CRISPR-Cas9?
Yeah.
That's like a molecular scalpel that allows scientists to edit DNA.
It's like a find and replace for our genetic code.
Exactly, and scientists are using this tool in some really interesting ways when it comes to HIV. One approach involves modifying that gene, CCR5.
That gene sounds familiar.
It's the doorway that HIV uses to enter our immune cells.
Oh, right. So by editing this gene, can we, lock the door, keep HIV out?
That's the idea. Scientists are working on disabling or altering CCR5, making cells resistant to infection.
That's amazing. It's like building a genetic fortress to protect ourselves.
It's a powerful idea, and early research shows it has potential.
What about that other approach you mentioned?
The other exciting area is putting antiviral genes directly into cells.
So instead of blocking HIV, we're giving our cells weapons to fight it.
Exactly. Imagine cells producing their own anti-HIV proteins, little virus-fighting factories.
Wow, next level stuff. Is this actually working?
Early stages, but the results in animals are promising, of course. Lots of challenges before using it in humans.
For sure. But it's an area to watch, it seems like. With gene editing, we're really pushing the boundaries of medicine.
Absolutely. And gene editing isn't the only futuristic technology. Personalized vaccines are really exciting, too.
Personalized vaccines, what are those?
Imagine a vaccine tailored just for you and your immune system. That's the idea.
So instead of one size fits all, we're moving towards, custom-made vaccines.
Precisely like getting a tailored suit, perfect fit for your immune system.
Love that analogy.
Yeah.
But how do they actually create them? It sounds really complex.
It uses some sophisticated technology. It starts by analyzing someone's immune profile, looking for clues about their strengths and weaknesses. Then they design a vaccine specifically to boost their response in the best way possible.
So it's personalized training for their immune system.
Exactly. By understanding how someone's system works, we can make vaccines more effective and targeted.
That's incredible, but this all sounds really expensive.
It can be, but as technology gets better and costs come down, personalized vaccines are becoming more feasible.
Exciting possibilities, but even with all this too, HIV is hard to target. It mutates so much.
Absolutely. HIV diversity is a big challenge.
How are scientists overcoming this?
Clever strategies. One is targeting those unchanging parts of the virus. We talked about the Achilles heel of HIV.
Right. Those bits HIV can't change without breaking.
Exactly. bNAbs are great at that, but scientists are also exploring things like mosaic vaccines.
Mosaic vaccines. I don't think we've talked about those. What are they?
Pretty ingenious. Imagine a patchwork quilt with pieces from different HIV strains. That's what a mosaic vaccine is. It combines parts from various strains to give broader protection.
So it's like showing the immune system lots of different HIV so it's ready for anything.
Exactly. And to make it even more high-tech, they're using computers and AI to predict which strains will pop up in the future.
So AI to stay ahead of the virus.
Exactly. It's an arms race, but we're getting closer to a safe, effective, and accessible HIV vaccine.
This has been incredible. I've learned so much about the science behind this.
Me too. It's been great sharing this with you.
And it's important to remember that even with the challenges, the progress is remarkable.
I agree. The scientists working on this are so dedicated and inspiring.
We've covered so much from the basics of our immune system to things like gene editing and AI. And I think the biggest thing I'm taking away from this is hope.
Yes, there's a real feeling of optimism in the field, despite how complex HIV is.
Scientists are making amazing progress, and I believe we will find a way to end this epidemic.
I couldn't agree more.
This has been an amazing deep dive into the world of HIV vaccine development. Thanks for joining us.